Now is a very special moment as we recognize the recipient of WORLDSymposium's 2025 Roscoe O. Brady Award for Innovation and Accomplishment. James Wilson, MD, PhD, has been a pioneer in gene therapy, his career spanning more than 4 decades. Jim did doctoral training at the University of Michigan's Medical School. His PhD thesis was entitled Human Hypoxanthine Guanine Phosphoribosyl Transferase, the molecular basis of the enzyme deficiency states. He then completed internal medicine residency at Massachusetts General Hospital, followed by a postdoctoral fellowship at the Whitehead Institute. Returning to Michigan, he was rapidly promoted in the faculty in internal medicine to professor and to chief of molecular medicine and genetics. Dr. Wilson was recruited to the University of Pennsylvania in 1993. Always laser focused on gene therapy, he created one of the first and largest academic-based programs in this nascent field. Dr. Wilson has authored more than 800 scientific articles. His teams have developed numerous FDA-approved AAV-based gene therapies with approximately 20 programs currently in development. While in academia, Professor Wilson has also been actively facilitating commercial development of new gene therapy platforms, including the establishment of 10 biotechnology companies. Thus, his efforts have contributed to successful clinical programs of in vivo gene therapy for rare monogenetic diseases developed in the corporate sector, including Glybera for familial lipoprotein lipase deficiency, Luxturna for RPE65-mediated retinal dystrophy, and Zolgensma for spinal muscular atrophy. Dr. Wilson has major corporate affiliations as founder and advisor for Scout Bio, for Passage Bio, and for IE Cure. This fall, after 35 years there, Jim left Penn to take leadership roles in 2 startups that he co-founded. He is now president and CEO of Gemma Biotherapeutics, which is focused on creating lower-cost therapy platforms for rare diseases, and executive chair for Franklin Biolabs, a genetic medicines contract research organization. The goal of both Franklin Biolabs and Gemma Bio is to translate scientific work into clinical trials to commercialize and to distribute new genetic medicines around the world to patients in need. This past October, Gemma Bio secured $100 million in funding from Brazil's Oswaldo Cruz Foundation. The purpose is to advance gene therapies within Brazil's public health system. The aim of this partnership is to create affordable gene therapies for rare diseases such as GM1 gangliosidosis, Krabbe disease, and for metachromatic leukodystrophy. I would like to stay clear of overused phrases in the current vernacular. However, in referring to Dr. Wilson, it is neither a casual trivial plaudit, nor is it hyperbole to characterize Jim as brilliant, and to recognize his diligence and accomplishments, and while still at mid-career, as truly awesome. Would you all please join me in congratulating the 2025 WORLDSymposium recipient of the Roscoe O. Brady Award for Innovation and Accomplishment. Thank you, Jim, for your past and future work.
Thank you, Jim, for coming to the WORLDSymposium 2025 to receive the Roscoe O. Brady Award, continuing his tradition of innovation and accomplishment. Congratulations.
Thank you very much, Chad. Well, I want to express my gratitude for Chet and other members of the community for this incredible honor. And the jobs we usually hold, we do not expect recognition, but only results. But but this coming from a community of my colleagues and the patients we serve is extra special. It is a very extraordinary time in the field of gene therapy for rare diseases, and it required extraordinary decisions, which I want to share with you. So I'll briefly comment about where I began my career in genetic medicines and rare diseases was during my graduate training at the University of Michigan, where I studied this disease, Lesch-9 syndrome, a horrific neurologic disease with a bizarre and destructive tendency for self-mutilation. And what became clear to me at the time, this would have been around 1980, how neglected these diseases were, but what the opportunity would be. And I, being a young man in which anything was possible, I thought about the notion of gene therapy. And from there, moved to Boston, where I did my residency, but it was really to seek out scientists who knew a lot about viruses so that we could develop virus as a delivery vehicle at the MIT under the tutelage of David Baltimore and Richard Mulligan. And so in those early days, we developed the notion of engineering a virus to deliver a gene into cells and to shuttle in therapeutic genes in a way in which could permanently change the phenotype of the cell and then the prognosis for the patient. There have been many different viruses that have been deployed over the years, but the approach that we've taken is to directly deliver the virus or vector into the patient to engineer the cells as opposed to engineering the cells outside the body. And so as I thought about my trip to San Diego, reflecting on my career, which has spanned 40 years in gene therapy, I'd have to concede and admit that there have been incredible successes. So there are now 8 approved gene therapy for rare diseases, prices ranging from $0.9 to $4.3 million. I guess you could say that we succeeded. However, let me just illustrate 3 different products in terms of the opportunity and the challenges. The first is treating an inherited form of blindness, which is super rare, and in which the revenues are quite modest, but it was the first. Then there were 2 others that are deemed commercial successes. One is to treat spinal muscular atrophy, a more common genetic disease, with a price tag of $2.1 million, but revenue for the company of $1.2 billion. And then very recently, a product for Duchenne's muscular dystrophy, in which a more common genetic disease with projected revenue in excess of $1 billion. So from a commercial standpoint, the SMA and the Duchenne's products were deemed a success, and probably, in the eyes of many, the only successes from a commercial standpoint. But even with those, there came a cost, and that was a high price for these one-and-done treatments. So as I also reflected on those 40 years and those successes, I also realized that there was not clearly a path forward for leveraging the experiences that we had over those decades. And in fact, I would say that most patients in need of gene therapies will not get access to these for 2 reasons. They are either too rare or they're too poor. Let's talk about being too rare. This is data I pulled from Human Genetics Journal about the distribution of rare diseases with respect to prevalence. Over on the left, most diseases are very, very rare, and on the right, there are a few that are not. And what we see is that the 2 successful commercial products, SMA1 and Duchenne's, are on the common side of rare diseases, sort of on the right side that could potentially justify an investment. But unfortunately, most of the genetic diseases, including virtually all that are the topic of this meeting, run the risk that they're below the bar and therefore may be too rare for an investment. Let's talk about too poor. These are data that I pulled recently from common databases for different countries in terms of the per capita spend on healthcare for individuals, with the United States in the lead of $12,000 a patient. But it rapidly falls off. For example, a country that we're working with in Latin America, the per capita spend is less than $1,000. One could envision why those that live in the United States could get access to these multi-million-dollar drugs. But equally so, you can imagine many of the countries in which the per capita spend is less will not get access to them because they're too poor. Despite these concerns that I know I had about the commercial reality of gene therapy, there were progress being made for a few years. And then right around COVID time, we hit the wall, in which there was a stampede away from developing gene therapy and genetic medicines for rare diseases. There's a term that was coined by some investigators who wrote an article. They call it the Great Abandonment. Since 2021, drug makers have shelved, abandoned, or rolled back the development of greater than 50 rare diseases. Most of these are gene therapies, and when you look at the article, 16 involve lysosomal diseases. So the question we asked ourselves, is this the world that we want to live in? Do we want to live in a world where we have solutions that could modify diseases, but because there are challenges of the business model that they're not going to proceed? And I would argue, I am not aware of any time in the history of medicine where we had solutions broadly applicable that weren't taken forward. So what we decided to do was, rather than try to change the world as a professor of medicine at the University of Pennsylvania, I made the audacious move to become a chief executive officer of a biotech company that's going to try to find a different model. I can promise you one thing: when I finished my postdoc at MIT, I never envisioned or never aspired to be a CEO of a biotech company, but here I am today. And so what I'm going to describe today are our views on what is necessary to overcome this logjam, this sort of blockade of being too rare or being too poor. I am confident we can do it, but it's going to require we think out of the box. We have to innovate, not just innovate in technology. We have to innovate in business. We have to innovate in terms of how we regulate these products. And I think innovation will be the solution. So when we thought about putting together an effort that I would welcome others to participate in and to develop parallel approaches, it's all about innovation. And in drug development doesn't necessarily foster innovation, because there are very tried and true pathways and practices. So we have 5 pillars of innovation. One is to think about developing products not as individual products, but as platforms. Innovation in technology and manufacturing always helps. We should not stop. But as we've thought about a path forward, we think about innovation commercially. We think about how to build coalitions internationally. And then how we finance or resource this, we have to think about different ways of doing that. So let's start with the notion of a platform rather than a product. I would argue that where we are with lysosomal diseases, that this could become a paradigm for personalized medicine. And what do I mean by personalized medicine? Well, here's an example of a delivery vehicle, a vector, in which there's a gene, a therapeutic gene, to treat a particular disease. Is it possible that all we have to do is swap out the gene for another disease, swap out another gene for another disease, and so it goes, keeping everything else the same? This is a platform. This is personalized medicine. Lysosomal storage diseases are exquisitely positioned to develop this kind of approach. Now, if we can, what are the criteria of a platform and what are the advantages? We have to keep everything else the same except for the gene. Common disease pathologies, common target cell, the same delivery vehicle, the same route, the same dose, the same manufacturing, the same, but swap the gene out. And the advantage could be that the chances of success in a platform could be much greater if you've already succeeded in the platform, and we've already shown that. That we could leverage that experience and spend less money. It's about spending less money so the cost of drug development is less, so that we can afford the development of these products, make them available, and possibly even charge less. And we can accelerate timelines because time is money in business. But where I get the biggest pushback, and trust me, I have through 40 years, people telling me you can't do that, is in a commercial model. But I think that there's something special about rare diseases in 2025 and one-and-done treatments that could create an opportunity for us to develop new commercial models that are much more affordable and much simpler and much accessible. Because traditionally, not always, the paradigm is biotech develops a product, commercializes to pharma, or licenses to pharma, and pharma commercializes. But I don't think pharma is going to be there to take the baton. In fact, there's been a pretty significant retreat over the last few years, at least for one-and-done treatments. But there's something special about rare diseases and these gene therapies or genetic medicines that would allow us to think differently. We believe that the way that these products should be delivered is through centers of excellence, not every hospital, not every pediatric hospital. That they will serve as regional hubs for patients that would then travel to the center of excellence to receive their treatment and then to go home. Follow-up can be provided by referring physicians. When I present my budget to investors, they ask me, what is the sales and marketing budget? And I said, well, we don't have one. And they presume I'm some stupid academic that doesn't know what we're doing. But we probably will have some spend, but we're going to rely on partnerships and stakeholders with the physicians and the scientists and the patient advocacy groups to work collaboratively and to use your networks and as well as the use of social media to preclude that requirement. And then establish these centers of excellence and these hubs, not only in high-income countries, but around the world to assure global access. So we're working in the United States through the company that I started, which is located in Philadelphia, with networks in many of the hospitals that are represented here. We've really leaned into Latin America, and I'll tell you about that briefly, spending a lot of time in trying to establish a similar structure in the Middle East. So let's start with our work in Latin America. 211 million people in Brazil, 633 million throughout Latin America. Incredible founder effects, burden of genetic disease high. Per capita spend in Brazil, $721. So the government of Brazil reached out to me, the Ministry of Health, and asked the question, can we partner together to make drugs more affordable, gene therapies more affordable for patients in our countries? And let's do it. So a startup biotech company that essentially had no money, was looking for money, run by an academic, we were able to consummate a collaboration with the Ministry of Health where they provide us $100 million of non-investment money to help us develop our products, in which we then in return provide them a license to these products so they could distribute under their public health system these drugs at a much more affordable cost. You may say $100 million is a lot. Trust me, the one in charge of our budget, that is a lot, and it's incredibly helpful for us. But when you think about the cost of these multi-million-dollar drugs over 10 years, If we could provide them at a much more affordable cost, it could save the country a significant amount of money. What's equally exciting is how can we build on this presence in Brazil throughout Latin America? And stakeholders have reached out to us, and we're in discussions with FDA, with Gates Foundation, and with many local stakeholders to try to integrate and coordinate these activities in Brazil through the many other countries in Latin America. A similar work stream is underway in the Middle East. And then financing. Traditional way that you finance a biotech company is through venture capital. My experience, walking around hat in hand for the last 2 years, is there is some way to raise money through venture capital, but for us it's not enough and it's not sufficient. So we've really pursuing a mixture of financing strategies, and I would argue that others should think about that as well, in which an investor is not just someone who gives you money. An investor is a partner, a strategic partner, such as Brazil, such as what we're going to develop in the Middle East, and also foundations. So foundations have approached us to try to work with us to raise the capital necessary so that we can move that bar about being too rare even further to the left. And there are many creative ways in which foundations can do this. And if we have any assets, such as intellectual property assets, license those. So I want to talk about our work in lysosomal storage diseases, because this has formed the basis for the work that we're doing in the company. So we have a CNS platform for treating diseases that affect the brain. And this is our platform. We have an AV9-like capsid. We have a standard manufacturing process. Through our collaborators at Penn, we've developed a way to inject a vector that goes throughout the central nervous system in this compartment like a lumbar puncture, but it's below the occipital bone. It's called the cisterna magna. We've trained physicians in 12 countries, 13 companies we're helping with. And we've evaluated this in 541 non-human primates for safety and also for demonstration of durability and efficiency. And throughout the brain, we can get consistently levels of stable, safe gene expression at about 5%. And this is just an example of work that we did in non-human primates. On the top here is a low magnification view throughout the cortex of a non-human primate, where the gene expressing cells are green. And you can see that there's sort of collections of cells that go throughout the brain, but it's not 100%. And below it shows that many of these cells are neurons, but also other cells take the gene up. So the question is, when is 5% gene transfer enough to treat a global neuropathic disease? The answer is when there's cross-correction. And this is where many diseases that are the topic of this meeting were positioned very well for us to take this approach. As you know, there are many of these diseases are multi-organ. Enzyme replacement can address very adequately in some diseases, less so in others, the somatic, the non-CNS component, but it is the CNS neuropathic aspect that we're trying to address with our CNS platform. I'm sure all of you know about cross-correction, but just to remind you that if we have a bunch of diseased cells and we get a vector into one cell, that'll fix that cell, but that cell will also make enzyme that will be taken up by adjacent cells, so that you then end up with corrected cells beyond those that take your vector up. So it's really one of the primary reasons that we decided over 10 years ago to focus on neuropathic diseases with cross-correction. So we started with MPS I, and I've often been asked why that's the case, because we now have 6 programs in the clinic and we have 5 others preclinically. And the reason is I met my colleague and friend Mark Haskins, who many of you know well, who had these incredible large animal models of storage diseases. And in MPS I, he had a dog model and he had a cat model. My experience is it's pretty easy to cure a mouse, but if you can actually cure a dog or a cat of the disease, that de-risks the program. And this just shows the clearance of storage abnormalities, both GM3 and LIMP1, in both the dog and the cat. This is normal, MPS 1 and AAV treated. Now, there isn't a non-human primate model of storage disease, but we did do experiments in infant non-human primates, followed them for 4 years, and were able to show after 4 years the levels of stable gene correction were actually remarkably high. I think this may have to do with the fact that our efficiencies are higher in newborns or in infants, 6 to 29%, 2 to 13, and 6 to 26. So we've, over the years, have rolled out 5 programs in the clinic. First with a company I started, Regenix Bio, which is Hurler syndrome, which is completed phase 1/2 and pivotal studies. Hunter syndrome, the same platform. They're submitting a BLA now, hopefully approved in 25. It will really be an important validation of the platform. Regenix Bio is attempting to try to license these products so that they're developed throughout the world. I started another company after Regenix Bio passed on other programs, and this is a company called Passage Bio for GM1, Krabbe, and MLD. We did all the work. GM1, we're now in discussions with health authorities about what the next steps are, Krabbe and MLD, in phase 1/2 studies. We have 5 other programs ready to go, locked and loaded, ready to get into the clinic and manufacture, and I just want to show 2 of them. One is MPS 3A, and this is a mouse model where we show complete survival and, importantly, correlating with a reduction in heparan sulfate in the CSF. MPS 3B, same thing, shows survival in the mouse model, directly correlates with a correction of CSF heparan sulfate. And because of the efforts of this community and others, we've convinced health authorities that this could be a path for accelerated approval. So I'm just going to end on a final pitch, and this is a pitch to you all. I believe that the kinds of many of the diseases that are the topic of this meeting are best positioned for commercial success, not the least position. So let me describe why. As I've thought about 40 years of experience, not only in our group but others, what is necessary for a commercial success? There are a lot of gates. There are a lot of barriers. In this, you've got to start from something that works in the clinic. You got to get approved. It has to be priced. Docs have to prescribe it, believe it or not. Some gene therapies aren't, and there has to be adequate sales. When I think about the diseases that we're studying, and many of you that you are studying, we got all of these nailed. We've got the technology, whether it's systemic or ICM. Approval of these products is much easier than many others because we have biomarkers, we have accelerated approval, great natural history data. The products will be priced if they save lives. Surely docs will prescribe them. The challenge is the individual markets. Everything else is a green light. And if we think about aggregating these programs so the aggregate market is sufficient, then I believe these are really the best position for commercial success. Now, obviously, I voted with my feet. I started a company to do this. But at the age of 69 with 3 grandchildren, I didn't take this one lightly. And we are planning for success, not because we hope, but we really believe that all the elements are there. So I do not want to miss the opportunity to really thank a number of people that have been so supportive of our efforts. In particular, my colleagues, Juliette Hordeau and Christian Hinderer, the scientists in the lab that have spearheaded these programs. Mark Haskins, my dear colleague at University of Pennsylvania School of Vet Medicine, taught me a lot about storage diseases. Monique Malloy, who's here, a professional partner, been working with me at Penn for 30 years. And Roberto Giuliani, who has just been a wonderful collaborator and had a lot to do with convincing the Brazilian government to work with this guy from Philadelphia, PA. Thank you, Roberto. But also, George Weiss and his family, Allison and Chip, who are here today, they introduced me to a member of their family years ago. And it was based on that interaction that we redirected our efforts to storage diseases. With their generosity, we started the Orphan Disease Center, which I led for 10 years and just stepped down, but proudly have raised $14 million and distributed that to 36 institutions and 59 PIs. But then finally, and most importantly, we want to say thank you to all of the patients and the advocacy groups that have been part of this journey. Our work has only begun, and we look forward tomorrow, next week, next year, to really turn the page and try to look forward, not in the rearview mirror, for a lot more successes. So thank you again, Chet and others, for this incredible honor, and appreciate the invitation. Thank you.